you for allowing me to speak here tonight. I'm really happy to be here and present in front of you all. So my name is Atreya Manaswi. I'm a high school student in Orlando, Florida in the 10th grade at Orlando Science High School. I do my honeybee research affiliation with the University of Florida and also the US Department of Agriculture. And I'm going to be presenting on nutrition management and pollen substitutes today. So before I get into my actual presentation, I'd like to begin with my backstory of really how I got into beekeeping at such a young age. So 4 years ago, I went on a short fishing trip with a friend and his grandfather. And while we were on the boat, my friend's grandfather began telling us some stories about his beekeeping practices. And one thing I remember was about how decades ago, he said he would get dozens of barrels of honey, but how that season he got merely 3. So this was really shocking to me, and I went home and I dug into the research and the statistics, and I did some brainstorming. And after that, I decided to conduct extensive research on honeybee conservation at the lab in the University of Florida, the honeybee research lab there. This is actually a picture on the top right of me in my first year of honeybee research in the 7th grade at age 12. So since then, actually, I'd like to add in the laboratory, it's a very comfy and cozy environment, contrary to the field where it's not air conditioned. It's super hot and you're lifting these heavy hives. So it really taught me about the value and the hardships that go into beekeeping and the effort it really takes for beekeepers to put food onto our plates. So it was really eye-opening for me. And after that 6th grade summer and 7th grade for that project, I progressed to these field trials, and that's really when I saw all this and it opened my eyes. So for my presentation, this is the basic breakdown for it. I'd like to begin with the importance of nutrition, focusing on some macro and micronutrients, moving on to pollen substitutes, what they are, how you can use them, and whether they're actually effective or not. And then we'll be analyzing some colony performance metrics and basing this on some research. And then I'd like to focus on my research specifically that I did at UF. And I'll be discussing the experimental design and then also the conclusions from that study. And then lastly, I'll provide some recommendations on nutrition management. So the key takeaways I would like you to have by the end of this presentation are firstly, what the different types of macro and micronutrients are that are needed by honeybees. Secondly, what a pollen substitute actually is and what it's composed of, how it's placed in the hive, and the current research on this field of study. And then thirdly, how you can apply my findings into your hobbyist or commercial beekeeping practices. So let's begin. Firstly, with the importance of nutrition, honeybees, like all animals, need many macro and micronutrients. And these include amino acids, which they derive from proteins, carbs, which use as a source of energy, vitamins, minerals, and then also water. A lack or imbalance of these factors can actually lead to malnutrition, which is an increasing threat as it comes together or synergizes with other factors such as deforestation or climate change, pests, parasites, diseases, and also pesticides. A variety of sublethal factors are also associated with this malnutrition, and these include susceptibility to diseases and also chemicals and insecticides. So let's begin with the first macronutrient, perhaps the most important, which is proteins, and the amino acids are what the bees derive from this. So bees obtain protein in the form of pollen, which also provides them with other things such as minerals, lipids, and fats. Now, amino acids must be acquired through these external sources, as the bees cannot synthesize them themselves. Now, pollen is processed once it's brought into the hive into a source called bee bread, which the bees store and use whenever needed. Now, insufficient protein can result in effects such as decreased brood rearing and shortened worker lifespans. For carbohydrates, bees use this as a source of energy. Now, carbohydrates are firstly turned into glucose, which is then used as ATP through the Krebs cycle. And ATP is essentially the fuel that cells use to function. Now, bees get their carbohydrates through nectar and honey, and nectar is the primary source for carbohydrates. Now, moving on to vitamins, minerals, and lipids. Nurse bees use a vitamin B mineral complex during brood rearing, actually, and vitamin C is also very important for this process. As other insects use high levels of magnesium, potassium, phosphate, and other such minerals, it's also thought that honeybees need these in high quantities for their overall homeostasis and body regulation. Now, an example of a major sterol or cholesterol that bees use is sterol 24-methylene cholesterol, and it's very common in pollen. And like amino acids, bees must acquire this as it cannot be synthesized. Now let's discuss what a pollen substitute actually is. So fundamentally, a pollen substitute is just an alternative source of nutrients, like I discussed. These macro and micronutrients that include proteins, fats, vitamins, minerals. And these are generally composed of carbohydrates, vitamins, oils, yeasts, honey, pea proteins, and other such things. So there are two main types of pollen substitutes, and they are those that are already pre-mixed in the solid patty form, and then the latter is the powdered form. This is the example of the first thing that I mentioned, the pre-mixed patty form. And so this is a solid that's placed on top of the frames in a super. So the bees can feed on this whenever they like inside the hive. And so this is something very convenient to use. You just place it in the hive and let the bees feed on it. The alternative to this is actually using a powdered form for a pollen substitute. And this is placed in the vicinity of the hive inside some sort of cup. So bees can feed upon this. They have to forage and bring that back into the hive. Now, why should you actually use a pollen substitute? As agriculture becomes increasingly monocultural, meaning that these plots, which were originally decades ago, there were many different types of plant varieties plotted on these. Now they've become commercialized so that a single crop species is placed on these huge fields. And this is a negative for the honeybees themselves, because honeybees being in the vicinity of those crop fields, they only have a limited source of pollen, and they need diversity in their pollen source, because this diversity and also quality is critical for their colony growth, their nutrition, and overall survival. So to ensure that colonies are healthy, beekeepers often use these commercial pollen substitutes as an alternative source to natural pollen when there is pollen dearth or the pollen quality is very limited or not diverse. Now, this begs the question, are pollen substitutes actually effective? So there's no clear-cut answer on this. Literature does actually show some mixed results, and the definitive answer here is that depending on the conditions in your actual beekeeping practices, it's best to use them or not use them. But one thing is for certain, when natural pollen is available in the area, bees will actually prefer to consume the natural pollen rather than the commercial substitute. And I also saw this in my research result, which I'll discuss in further slides. So there have been several studies in the last few years that have shown that between colonies fed these pollen substitutes and colonies fed no diet at all, a negative control, the colonies with the pollen substitutes did increase their adult bee populations and other colony strength metrics, such as their strength, their brood rearing, and other such things, and also foraging activity. So thus, a spring and winter pollen supplement could serve as an insurance almost when the weather is bad or there's a period of pollen dearth, or a faster spring buildup and also a higher honey yield. So it essentially serves as a backup in case there is pollen dearth or no other good nutrition available in the area. Now let's analyze some of the colony performance metrics, and I'm going to be looking at 4 here. So these will include adult populations and longevity, brood rearing, honey production, and the queen quality in rearing. And these 4 parameters are all in response to being fed these pollen substitutes. Let's look at adult bee populations and longevity. So the majority of the research on this topic, again, shows that adult populations did increase with the pollen substitute feeding compared to no feeding at all. It should be noted that these studies were conducted in unfavorable weather conditions, such as rainy seasons, harsh summers and winters, and pollen dearth. And the region is also thought to play a role, as these warmer climates show less of an impact on the feeding than these harsher, colder climates. So this really shows that in these more extreme conditions, the bees will do better with a pollen substitute, something to consume, rather than these warmer, more favorable weather conditions and climates. So let's look at brood rearing. The majority of studies on this topic show, again, that in extreme conditions, using a supplement does aid in brood production. So spring feeding of these substitutes can enhance brood production, whereas summer and fall feeding is not thought to be as favorable. In fact, some studies have shown that fall feeding caused brood rearing burnout, and the colonies actually ended up producing less brood in general. And this is actually because providing the bee colony with so many resources caused them to use this in an over expenditure, and they essentially started laying too many eggs, and this caused that rapid burnout, which ultimately led to the demise of a colony. Now, it should be added that the addition of pollen, soy flour, and lipid extracts in the 2 to 4% ranges, such as cholesterol, do aid in the supplemental diet feeding. So they have been shown to be beneficial when they're added to these diets. Looking at honey production, numerous studies have shown that pollen substitutes do increase honey production. Abbas et al. In 1995 even showed a difference as much as 3.8 to 4.6 times of an increase between controls and those fed these substitutes. Other studies on this topic, however, have shown that honey production and other strength metrics increased following a poor season with poor weather conditions that limited foraging activity in the previous season. So these results also further substantiate that the pollen substitutes will likely benefit the colonies after a poor season. And analyzing the queen quality and the rearing, there are very limited studies that have been done on this topic. There's only 3 really. The study conducted by Manning in 2018 showed that queens fed different diets, a 10% oil diet, a 5% oil diet, and a defatted soy protein diet. When the queens were fed these different diets, at the end of the study, they had different weights. Now, there was no further work done on the weights and their correlation to higher reproductive success, but Manning did show that feeding them different types of diets resulted in a different weight at the end of the study. And the study conducted in 2016, which was a queen longevity study, showed differences between the queen losses when feeding some colonies B Pro while feeding some other colonies Mega B. However, there was no negative control in this study. And the study from 1943 did not have a rigorous statistical analysis or a discussion, so not much can be concluded from that. So as you can tell, there's not much work that's been done on this topic. Now, moving apart from all of that, the clear-cut answer is that these substitutes are effective during periods of pollen dearth when few natural pollen resources are available for the bees to utilize. But my suggestion is only to use these when there is a need, because ultimately it can become expensive to use these pollen substitutes, so you should use them only during periods of pollen dearth. Now, research is still being conducted on optimal timeframes for feeding and nutritional practices, but as I mentioned, feeding in the spring is likely to be your best option. So let's move into my research now. So my research focused on a comparison of 3 commercially available pollen substitutes and wildflower pollen, and this was done in an in vitro setting in the laboratory. So in this study, there were 4 diets that were used: wildflower pollen, AP23, Mega B, and Ultra B. And AP23, Mega B, and Ultra B were the commercial diets that were used in this study, and they were compared to wildflower pollen. So there were 2 research goals here for this study, the first of which being to determine whether the pollen was actually more attractive than the substitutes, and the second was to determine which of these 3 commercial substitutes was the most attractive to the honeybees with the presence of the wildflower pollen. And reviewing the study design, as I mentioned, this research was done in an in vitro setting in the laboratory, and cages were used for experimentation. So on the left, you can see an example of a cage here. And so there's 4 holes here, which are for feeder tubes, as you can see in A. So inside this tube, a diet was put in, 1 gram of each diet. So there were 4 holes and 4 treatments. So for example, this feeder tube could have had 1 gram of pollen. This feeder tube could have had 1 gram of Mega B, 1 gram of Ultra B, and 1 gram of AP23. And so these were available to the bees at all times to feed. And this was a choice test essentially. Figure B here shows the sugar water feeders that were used to supply the bees with sugar water. And then C is the ventilation holes for the bees to breathe. Now, these cages were used in the experimental design, and there were 10 of these experimental cages that had 10 bees inside of them each. So in one of these cages, there was 10 bees. And so these bees would feed on the diet. And then there were factors that were used to measure which diet was actually the most effective, and I'll discuss that. So there were 10 bees in these experimental cages, and there were 10 cages, but there were also 5 evaporative control cages. So these evaporative control cages had the same setup as these normal cages, except they had no bees inside of them. So you have a set of cages with bees and the diets, and then you have a set of cages with everything else except the bees. And so these evaporative control cages were actually used to account for the evaporation. And that's important because these diets do contain a high lipid quantity. And so the lipids can evaporate. There's a lot of moisture in these diets. And because of that, they can actually lose weight due to evaporation. And so looking at this next slide, this is actually how it looks. Setup you can see here on the left is the evaporatives, and on the right is the cages with the bees, which are the experimental cages. And so, as I mentioned, two factors were used to assess for the diet preference. The first was weight. So, this was run over the course of a week, and each day a new diet would be put in, and then that diet would be taken out, and another diet would be put in. So at any given point in time in this cage, you had 4 diets, but every day these diets were replaced. And so before they were put in, the weight was taken, and then after they were taken out at the end of the day, another weight was taken. So you got an initial weight and a final weight, and so these were subtracted with one another. And the same thing was done for the evaporative controls. And so taking into account the differences between the cages with the bees and the cages without the bees, an actual weight was determined for what the bees consumed. And so this was one factor for seeing how much the bees actually consumed throughout the course of the study. And then the second factor was the observations. So bi-daily observations were performed in a red room setting. And here, I can describe it here. You can see this image on the left is a red room. And so bees actually can't see in red light. Therefore, they're observed in this setting. And so for 5 minutes on each of the 10 cages, a total of 50 minutes, I would observe these cages and look at how long the bees in that cage were interacting with each diet for. So let's say, for example, a bee was interacting with Mega Bee for 10 seconds. I would note that down. And then say another bee was interacting with Ultra Bee for 5 seconds. I'd also note that down. And then at the end of that observation for that cage, I'd sum up the totals and get a total number of seconds that the bees spent interacting with a specific diet. And so that was the second metric used. And the other metric that was the weight is shown on the right here. The weights were measured to see what the weight at the beginning of the day was before the bees were allowed to feed on it in the cage. And then at the end of the day, that same tube was taken out and then weighed again to see what the difference between that weight was. As I mentioned, the evaporation was also accounted for using the evaporative controls here. So these are the results. Looking at these graphs, you can see these were the 4 days throughout that week. And Mega Bee here on the first day, which is in green, seems to be doing the best. But throughout the rest of the study, Mega Bee diminishes significantly. Pollen starts off very low here, but it tends to increase and stay constant during the rest of the study. And this is actually, it could be ascribed to the fact that as the bees were growing, they actually needed more of this pollen, so they were consuming more, as there's a linear trend here, and they're consuming more throughout the course of this 4 days. AP23 also seems to be a little bit constant here. And this is another graph for the weight change. You can see AP23 is A, Mega Bee is M, Ultra Bee is U, and pollen is P. So this is the change in weight. And this is the change in weight in milligrams. So you can see pollen has the greatest change in weight here. It has the greatest negative number, essentially. So it dipped the most in weight. It lost the most weight, meaning that the bees consumed the most pollen throughout the course of the study. You can see AP23 here. It's slightly lower than Mega Bee, not significantly. And then Mega Bee, and then Ultra Bee. So it was shown to be that pollen was the most effective, followed by AP23, and then Mega Bee, and then Ultra Bee. This is the pie chart based on the total observation times. So you can see here, this is in seconds as a graph. Pollen is roughly 70% of all of the time in this graph. So looking at any given point in time at that cage, 70% of the time, the bees would be interacting with pollen. 25% of the time, roughly speaking, they'd be interacting with AP23, and then the other 5 and 4% were spent on Mega Bee. So this is another graph with the time spent feeding. So this is the seconds graph. You can see here, just like this pie chart, pollen is shown to be the greatest here, P, followed by AP23, which is in orange. Mega Bee is this third best, and then Ultra Bee is the least. So the conclusions of this study were that pollen was by far the most attractive diet amongst the treatment groups, and AP23 was shown to be the most attractive commercial substitute in this study. So I believe future research on this could replicate this in colonies to see if it yields similar results, and also performing experimentation during periods of pollen dearth and in seasons of availability would be interesting as it would yield different results based on the bees' preferences in the season. These are some additional resources on nutrition management that I will share, and these can be circulated amongst you guys if you'd like to take a look at these. These are very helpful. The last link here is the UF Honeybee Lab page. This has some good videos on beekeeping, just in general, and these are all other PDF documents that you can take a look at. They have further information on nutrition. And that's all I have for you today. So thank you so much for your time and attention, and I'm open to questions now.
Wonderful. That was great. You did a good
Thank
job.
you.
Rich, you have any questions?
You did say, I did notice up here that I noticed that there's not a lot of pollen coming into our hives right now up here.
Yeah.
And the honey production was down too this year. So my thought was on ours, I'm going to actually put pollen patty in. I actually use the AP23 patties, but I'm not going to put a full patty in right away. I'm just going to put like maybe a half a patty in and see how well they take it. So I just don't see a lot in my hive. There's some pollen in there, but there's not a lot coming in. So I thought we'll try to boost them up, and maybe you're gonna have to do a little sugar water on them just to boost them up a little bit. So.
I am in the same boat with that. I didn't see hardly any pollen in my hives. And I've been feeding them sugar water, and they drink 8 gallons a day. I've been just feeding them as much as possible.
Yeah, that sounds like a good course of action. I'm happy to hear that you're using AP23. That's actually the one that I found was the most attractive of all of these in the study. So that's great to hear. One thing you might want to be careful about, I'm not sure how bad this is actually related to hive beetles, but when you put those pollen patties inside your hives, sometimes if the colonies are weak especially, those pollen patties tend to be a really good place for hive beetles to reproduce. So you might want to be careful about, how much of the pollen patty you put in, and using half of it and seeing how the bees respond is a good strategy.
Cool. Very cool.
I'd say, Madison, be careful. They're feeding all that sugar water so they don't get all honey bound yet on you.
I know.
Don't get too much in there. So
I know it.
Still want brood yet for a while.
Yep.
If anybody has any questions, I'm happy to take them in the chat, or you could also unmute if you'd like. Anybody who's in the meeting.
What is your contact information? If anyone has any questions for later.
Sure, yeah. It's actually on this slide, this email. I can also drop it in the chat.
Perfect. Excellent. Cool.
Okay.
I suppose if you would like to, I guess we don't have hive beetles here, but I know Southern Iowa didn't have hive beetles either, for till the last couple of years. So I guess you want to go through it. We, might be interested in learning a little more about them and how to control them too, because, eventually they might be up here too. So
yeah.
Sure. Yeah, I'm happy to do that.
Hi, this is Emily, and I have had hive beetles in the past. It was when my hives were in an area that was too shaded, and they got in there, and it was disastrous. But, but that was the only year I've ever had trouble.
Okay, cool. Okay, Rich, do you got any ending comments? Do you have anything else for us? Any events coming up? I don't know. I don't have anything on my calendar.
Well, I know. Well, the biggest thing will be coming up probably will be Iowa Honey Producers has their annual meeting.
Oh, okay.
In November. I couldn't tell you the date offhand.
I can look it up.
Yeah, but that's coming here in a couple months. So that would be people are interested in it. There's supposed to be a very good speaker. Oh, I can't remember. Randall. I think I'd have to look back, but he's supposed to be very good. So I think he was supposed to come this last year, but it just didn't work out. So
is it Randy Oliver?
Could have been.
Okay.
Maybe it might be. I'd have to go back and actually look on it.
Okay.
But it's supposed to be very good. So, and I know it's going to be in Ames this year. Is where the annual meeting is going to be. I do know they talked about it's going to be in Ames. So not so far to drive this year. So.
Yep.
Sir, this PowerPoint presentation, it's about 35 slides, and it lasts for about like 45 minutes to an hour for the small hive beetles. I think I do have some resources on it at the end. Would you mind if I share those? I could drop those in the chat. There's some very helpful PDFs on control methods and then also biology. Is it okay if I share those with you?
That'd be fine.
Yeah, absolutely.
Yeah, and if you want to go through it kind of quick and just hit the high points, maybe that's fine
sure.
too.
Yeah.
Okay, sounds
good.
Go
ahead.
Because like I said, we don't have a lot up here right now, but I'm sure that we have a few. So.
Okay. Okay, so this is the PowerPoint. I'm gonna skip over to here. So this is some information on the biology of the hive beetle. And so these are actually endemic to Africa. And so in the 1990s, they actually moved over to America. In 1996, the first reports came out of North Carolina. And then in 1998, they were reported in Florida again. So in Africa, they actually pose very little threat to the African honeybee, but since they came to the US, they've been wreaking significant negative impacts on the European honeybees. And so these beetles are described as an opportunistic scavenger, really, and they destroy honey and pollen stores and damage cappings and comb. And this ultimately stresses the colony. So this image on the bottom right is a picture of the larvae in the colony. And so they tunnel through the comb and the pollen and the honey, and they defecate and everything, which starts fermenting. And so this is a life cycle of the beetle: the egg, the larvae, the pupae, and the adult. And as I mentioned, the larvae are really the main cause for this problem, and they appear very suddenly and in large numbers. So there's this yeast in their feces when they defecate that causes the fermentation of the pollen and the honey. And the brood rearing actually ceases, and the population starts to dwindle, and the hive may abscond, the bees in the hive. And this is termed as a slime out. So some interesting behavior between the honeybees and also this beetle is the bees will actually jail the beetle inside cracks and crevices in the hive. And so the beetles will actually sometimes fool the honeybees into thinking that the beetles are honeybees by taking their antenna and antenating against the mandibles of the honeybee, which actually confuses the honeybee into thinking that that beetle is a honeybee. So this causes this honeybee to regurgitate food and start feeding the hive beetle. So they're very smart. And this happens only about 10% of the time. The other 90%, the bee notices what's going on. But that's something very interesting to notice. So some general guidelines for small hive beetle infestations are just when combining or exchanging the comb to be careful. So you can introduce small hive beetle eggs from outside colonies inside the hive. Also removing old frames or hives because this makes for a very good spot for bees or beetles to deposit eggs. Also, when pulling honey from the colonies, it's important to extract within a 2-day period, as the beetles will often consume the wax cappings. And so they'll start defecating in the honey, and this renders it unsuitable for consumption. Another easy thing you can do is exposing your hives to more sunlight. So the beetles absolutely hate the sunlight, and the bees don't mind as much. They will get a little bit aggravated, but it's a good way to keep the hive beetles out. And then also when creating nukes or adding new supers or frames, it's important to only add as much as the colony can take, as the beetles will go and start wandering out in the hive, and the bees won't be able to patrol them. So there's these chemical treatments that are pretty popular for hive beetles, the checkmate strips, which are Comophos, and then guard star permethrin, which is a soil drench. And so you also have non-chemical treatment agents. This is apple cider, which is commonly used in these beetle blaster traps as an attractant. So it's placed in between the frames in the hive, and then the liquid inside attracts the beetles already inside the hive, inside that trap. It lures them, and then they drown in that solution. Another trapping technique or killing technique for controlling is diatomaceous earth. So this is placed in the vicinity of the hive, and it actually dries out the cuticle of the hive beetle. Another biological control method is nematodes. So these nematodes are known as entomopathogenic. And what that means is that they go inside the gut of the hive beetle actually and start consuming inside there and feeding off of the hive beetle, which ultimately kills it. So coming to my research, what I did was 2 years of research on hive beetles. The first year was where I tested 7 organic and inexpensive small hive beetle treatments. And the second year was where I developed a treatment based on beer, which was the best of the 7 treatments from the first year. So basically I tested 7 organic agents. These are the agents. And I tested these because they're all odorous, and this is based on prior study findings. And so they were placed inside these mini beetle blaster traps inside the hives and left in there for weeks to see what they would capture. And so each week I would go into the hive, place a trap, and then extract it and see what the hive beetle capture was for that trap. And so this was done for a 6-week period with weekly data collection and trapping. And so after my data collection, I ran an extensive stats test. And through this, I found that mango mix, cantaloupe, yeast, and beer were all more effective than apple cider when compared with just apple cider and that treatment. So these 4, mango mix, cantaloupe, yeast, and beer. But this did not tell me which of all of these 7 groups was the most attractive. So to do this, I ran an ANOVA test, and this would tell me if one group was significantly better than all of the other groups. And so I got a yes for this. And so there was at least one significantly different group, which is what my test was saying. But this specific test didn't tell me which of these groups was the best. So to actually do this, I followed it up with this test called the Tukey HSD statistical test. And so this test told me that the beer was the most effective of all of the treatment groups. You can look at the Pareto chart here and see beer in deep blue and see how it's much higher than any of the other treatments in this graph, which shows the efficacy of the treatment throughout the course of the 6-week study. You can also see the X, which signifies the mean for beer here in dark blue, and it's much higher than any of the other treatments, showing greater consistency in the beetle capture for this treatment. So ultimately, what I found in this first year was that beer was 33 times more effective in beetle capture than the apple cider vinegar, and it was the best treatment by far margin. Moreover, beer costs merely 5 cents per hive on average, and it's also widely and readily available. So looking at this, if you didn't believe me on those costs, you can see this is just from Walmart.com. Beer here costs merely 7.3 cents per fluid ounce compared to apple cider vinegar, which is nearly 20 cents. So beer is 3 times cheaper and also 33 times more effective. Now, I won't go too much into detail with this phase 2, but essentially I was developing a novel blend based on beer's chemical composition. So our goal for this was to create a refinement of beer in the form of a more concentrated solution. And the goal was to make this solution even cheaper and more effective than beer itself. And so this was done by extracting the chemicals from the beer and then running them in an instrument called a GCMS. And so this GCMS produced a reading, which was then analyzed. And the chemicals essentially that have a lighter weight or are more carried more easily in the air were identified based on this reading and then used in a blend. So those individual chemicals were tested in something called an electroantennography. So this is a test that sees how the antenna of the beetle is responding to these chemical compounds. So the antenna is excised from the head of the beetle, placed onto a forked electrode, and then the neurological response of the beetle is tested based on this chemical compound. And so the chemical compound is either read as a positive or a negative, and it's analyzed that way on the graph. And then based on those results, 2 blends were formulated and tested in the field. So they were ran on 28 different honeybee colonies for a course of 8 weeks. So it was an extensive field trial. And then after that data collection and the weekly trap counting for the beetle counts, these statistical tests were run. So the ANOVA was run once again, and a significant difference was determined. So at least one group was much better than the other groups. And then this was followed up with a post hoc test to see which of these groups was actually the most effective. And the oil blend was shown to be the best treatment by far. Here you can see the Pareto chart, which is a visual display of this. The oil blend in gray is shown to be much better than any of the other treatments. And beer is here. The control is mineral oil, which you can see here. So the conclusions from phase 2 were ultimately that this oil blend treatment was much more effective by a far margin than any of the other agents. And this oil blend was shown to be 5 times more effective than beer and half as expensive. Moreover, it was shown to be 7 times more effective than the mineral oil. So once again, you can see the radical cost difference here. Beer is 3 times cheaper than apple cider. And so the key takeaways for small hive beetle management are really to keep a good apiary, keep it clean, have sunlight exposure, be careful with how many supers you're placing on top of your hives, also how many frames you're adding, and to use whether it be chemical or organic-based treatment methods to treat for small hive beetle infestations. And it's the synergy of these two that ultimately reduces beetle populations to a low and more manageable level. There's really no silver bullet method for small hive beetle control, but combining these two is the best alternative. So these are also some resources for small hive beetle management, which I can share. And that's all for this presentation. So once again, if anybody has any questions, please feel free to unmute or send them in the chat.
I know up here, I've seen a lot of Facebook things go on, and they'll use an unscented dryer sheet and put it in like the corners of the box, and that will catch the small hive beetle in them. So,
yeah, I have heard of.
I haven't seen it work myself, but I was just told that's what they do, and it must be working somewhat anyway. So.
Yeah, I've heard about that. I'm not sure with that field of research or how effective that method is, but I have heard about that with the small hive beetles. A lot of people will use in-hive traps as well, using some sort of oil or something like that. So my focus is really trying to see what solution in that beetle blaster trap makes it the most attractive to these beetles. And I found that it was beer.
Nice.
That's readily available.
Right. So I was using Miller High Life beer. And so that was the ultimate conclusion from the first year study, really, that beer is the best organic agent that I tested.
All right. Cool. All right. Anyone have any questions?
I know, Emily, you talked about when you had them, you had your bees in the shade a lot, and you talked about, the shade is one place. So I do know when I put my hives out there, I want to make sure I get as much sun as I can. And if there is shade, I want to make sure I'm shaded, late afternoon or, something like that, but it's the heat of the day by then. So that's what I do with my hives.
Right. Well, we definitely moved them out of that area, so they are not in the shaded at all. So, and I haven't had trouble with it since.